2 hours after a single cup of blueberries. Researchers can measure the difference in how your brain retrieves a word from memory. Not weeks, 2 hours. And the compound responsible isn't fiber. It isn't vitamin C. It's a pigment, the same chemical that gives the fruit its dark blue color. It has a name most people who eat blueberries every week have never heard: anthocyanins. You've probably had them this morning on top of yogurt, or blended into a smoothie, or thrown into a bowl of cereal. The brain-berry capsules people pay $40 a bottle for? Many of them are trying to isolate this exact molecule, and most of the people eating blueberries every week are getting almost none of it because of what happens to the fruit before it reaches their mouth. Everyone already knows blueberries are good for the brain. It's the one thing you'll hear from a dietitian, a fitness influencer, and your grandmother in the same week, and they're all correct. The reputation is real, and it's earned. The problem is that the reputation stops at the word brain. Nobody who repeats it can tell you which part of the brain, which mechanism, which compound, which dose, which form of the fruit. It's become one of those wellness cliches like "eat the rainbow" that everyone accepts and nobody actually investigates. Meanwhile, an entire supplement category has been built specifically to isolate what's inside a blueberry. Walk into any pharmacy and you'll find capsules labeled Brainberry or Cognitive Support with anthocyanin extract printed on the back, $40 to $60 a bottle. Marketed to college students, night shift workers, people over 50, all of them selling a compound that's already sitting in a punnet in your fridge, if you're lucky, because there's a layer to how this fruit works that nobody talks about. And once you understand it, you'll realize the difference between blueberries that actually change your brain and blueberries that are biochemically closer to decoration. Here's what's actually happening. When you eat blueberries, anthocyanin metabolites get absorbed into your bloodstream and eventually reach your brain. Two things then happen inside your skull that nobody puts on the label. The first is a change in blood flow. Human intervention studies using brain imaging have found that within about 2 hours of consuming a blueberry drink, participants showed measurable changes in cerebrovascular function, more oxygen and glucose reaching working brain regions, correlated with faster reaction times on cognitive tasks. Think of your brain as a city that runs on delivery trucks. Every neuron is a shop that needs constant restocking of oxygen and glucose to fire properly. When those delivery routes narrow through age, poor circulation, chronic inflammation, the shops at the edge of the city start closing early. That's what a brain fog moment actually is, a word you should remember, sitting there in the dark because the truck didn't arrive. Anthocyanin metabolites appear to widen the routes and keep the trucks moving. The second effect is deeper. A study published in PLOS One, working with anthocyanin dosing in animal models, showed that supplementation specifically increased levels of something called BDNF, brain-derived neurotrophic factor, inside the hippocampus, the region of the brain most involved in forming new memories. Human trials on blueberry drinks have shown circulating BDNF is maintained or elevated after consumption, where the control group's numbers decline through the day. BDNF is best thought of as fertilizer for your neurons. It keeps brain cells healthy, strengthens the connections between them, and in the hippocampus, it supports the growth of new neurons, one of the very few places in the adult brain where that still happens. Low BDNF tracks with slower learning, weaker memory, and higher rates of depression. Anthocyanin metabolites are one of the few dietary compounds shown to move it in the right direction. The label says blueberry. The biochemistry says BDNF signal and cerebral perfusion enhancer. Nobody prints the second one on the punnet. If this is catching you off guard the same way it caught me when I first pulled up the research, take 1 second, hit like on this video and subscribe. This channel exists to dig into the science nobody puts on the label. And the second compound, the one that gets almost no attention outside of the research community, is what actually explains why long-term blueberry eaters look different on brain scans. Now the second compound, this one has a name almost nobody uses outside of research labs: pterostilbene. Chemically, it's a close cousin of resveratrol, the compound everyone talks about when they talk about red wine. But pterostilbene has a property resveratrol doesn't have. It crosses the blood-brain barrier easily and stays in circulation much longer. In practical terms, that means more of it actually reaches your brain, in a form your neurons can still use. What's it doing in there? 2 things worth knowing. First, it acts on your brain's resident immune cells, the microglia. When microglia become chronically activated, they release inflammatory signals that quietly degrade the connections between neurons. This is one of the mechanisms researchers now suspect drives the slow cognitive decline people notice in their 40s and 50s. Not a dramatic event, just an inflammatory background hum that never turns off. A study published in Scientific Reports demonstrated that pterostilbene reduced markers of this inflammatory response in brain endothelial cells, and other in vivo work has shown reduced microglial inflammation after pterostilbene treatment. It's not doing anything dramatic. It's turning down the volume on a process that shouldn't have been running that loud in the first place. Second, pterostilbene supports mitochondrial function inside neurons. Your brain uses roughly 20% of your body's total energy, despite being only 2% of your weight, and every one of those energy demands is met by mitochondria inside neurons. When mitochondria get sluggish, thinking gets sluggish. The research on pterostilbene consistently shows it activates cellular pathways that keep mitochondria clearing out damaged components and producing energy more efficiently. Add these effects together: better blood flow from the anthocyanins, more BDNF in the hippocampus, reduced neuroinflammation, cleaner mitochondrial function, and you're describing a fruit that maps almost one-to-one onto what the entire nootropic industry is trying to build in a capsule. Except the fruit has been in the produce aisle the whole time. Here's the layer no one talks about. Most of the blueberries the average person actually consumes aren't fresh. They're baked into a muffin, stirred into a batter, simmered into a jam, boiled into a compote, freeze-dried into a cereal cluster, and left on a shelf for 8 months. And every one of those processes destroys some of the anthocyanin content, the exact compound this entire video is about. A study using high-performance liquid chromatography analysis measured what happens to blueberry polyphenols after standard cooking methods. Baking retained roughly 74 to 76% of the anthocyanins. Boiling took losses as high as 47%. Microwaving hit almost 42% loss on the worst end. And when blueberries are cooked into recipes with baking soda, pancakes, muffins, quick breads, the alkaline environment destroys anthocyanins the fastest of all. Jam is the single worst preparation on record. The label still says contains blueberries. The biochemistry says half the reason you were eating them is gone before the plate reaches the table. Your habit was never the problem, the recipe was. So what do you actually do with this? 4 rules. Simple, non-negotiable. 1. Eat them fresh or frozen, not baked. Frozen wild blueberries tend to hold their anthocyanin content well because they're flash frozen at peak ripeness, and the freezing process locks the pigment inside intact cell walls. Fresh works when they're actually fresh. If they've been sitting in your fridge for a week, you've already lost a meaningful share of the compound. The 2 forms to avoid, in this order: jam, then muffins. 2. Aim for about 1 cup a day, 150 to 200 grams. This is roughly the dose used in the acute cognition studies, around 140 to 250 milligrams of anthocyanins, that produced measurable cognitive changes in a 2 to 3 hour window. Less than that and you're relying on cumulative effects over months. More than that isn't harmful, but you're past the point of diminishing return for the acute mechanism. 3. If you want them cold and raw, that's the top form: mousse, overnight oats, yogurt bowl, smoothie. If you insist on cooking with them, keep the temperature as low as possible and never combine them with baking soda in the same recipe. The alkaline pH is the single fastest way to destroy the pigment. 4. Pair them with something your brain uses: full-fat Greek yogurt, oats, or walnuts. Anthocyanin metabolism improves when the fruit isn't consumed as an isolated sugar spike, and pairing gives your neurons a steady fuel supply while the pigment metabolites work. Blueberries with yogurt or oats hit the brain very differently than blueberries eaten standalone on an empty stomach. Do those 4 things and you're getting what the research is measuring. Skip them and you're eating a garnish. You're not someone who just eats blueberries anymore. You're someone who understands that anthocyanin metabolites move measurable levels of BDNF in the hippocampus and support the cerebral blood flow that keeps your working memory awake. You're someone who knows why pterostilbene crosses the blood-brain barrier when its more famous cousin barely can. You're someone who reads a muffin recipe and sees an anthocyanin funeral. You know the difference between the version of this fruit that changes how your brain fires and the version that's just decoration in a $6 bakery item. Next video. There's a fruit sitting in almost every fruit bowl on Earth that carries more of a specific anti-inflammatory compound in its skin than any commercial supplement can affordably isolate. 9 times out of 10, people throw away the exact part of it that carries the compound. Watch what happens when they don't.